impressed about how many people are here. When I first was asked to come, I really thought there might be 20 people, and so to see this is phenomenal, and I'm really impressed. So I've actually had a long-standing interest in fatigue, and probably for 25 years I've been working in this field. And the problem with fatigue, of course, is that people, when they think of fatigue, they often think of on the right-hand side that someone is just they can't. Their muscles give out or whatever, but I think anyone that suffers from fatigue knows that that's not really the problem in PBC. It's not a muscle problem. It's a problem where it's coming from the brain. So I spend a lot of my time dealing with brain stuff. So I'm going to talk about brain stuff today. So if I'm not talking about the liver, it's because fatigue really is a brain issue. So the first thing is, why would a liver disease, so you have inflammation in your liver, why does that make your brain change? So I've spent a lot of time on that in my lab. I actually have a basic lab. We do animal-based research on that. And so there's different ways that the liver could actually signal the brain. It's not just the liver, it could be any organ. One way is it could release stuff into the blood, which then floats up to the brain. That's one way. Another way is it could activate nerves that come from the liver, and they go up into the brain and change the brain. And then we've identified this other pathway, which I've circled in red there. It's called the immune cell-mediated pathway, where actually immune cells, so white blood cells, come out of the liver or are activated in the blood, and they actually go to the brain and they go into the brain. And those white cells actually change how the brain works. So I think there's a lot of opportunities to potentially target that therapeutically in the future, because there are medications that are available that can treat that. But so that's mice, and this is man or woman, and so really, it's always interesting to look at animal models to get ideas, but really in the end you have to go to patients and go from there. So we've done a lot of work on using what's called functional MRI. So when people think of MRIs, they think of something that looks at structure, like they look at house or something like that, they look at an MRI picture on a screen or whatever. And that MRIs are quite good at that, but with that same technology, there's an MRI machine, you can actually extract data out of that, and that data is quite different. It can be, you can actually look at how nerves are activated in your brain. So you give you give them a task or something, and you watch how nerves get activated, and then you can watch how different parts of the brain talk to each other. So when we're not doing anything, we're just lying completely still, our brain actually talks to itself. And so when the areas of the brain that talk to each other, those are called neural networks. So just like any kind of network, they're in communication all the time. And so we are very interested to see, in patients with PBC, do they have a disruption in their neural networks, in how their brains talk to each other? This is the only image I'm going to show, and it doesn't really matter what it says, actually, but really what the bottom line here is that looks like an MRI picture of the brain, and that's what they look like. But this is actually, that MRI there is just as as a representation of the brain. In fact, this is information that comes from a whole group of PBC patients that we've studied in Calgary compared to a whole group of controls. And what it looks at how the brain differs and how it talks to itself between that whole group of PBC patients compared to the whole group of controls. Right? So this is not just one person. And the colors essentially represent, the blue is increased connectivity, which means that those neural networks are talking more to each other. They have a stronger connection. And the red colors indicate areas that have lower connection. And it doesn't really matter what areas are all connected, but the bottom line is that the PBC brains are very abnormal. And this is between all the patients. This is not one person in isolation. This is a consistent finding amongst all the PBC patients. And I just happen to pick this one because actually the neural connectivity changes between fatigued and non-fatigued patients. So there is a neurological basis that drives fatigue, right? Because the problem with fatigue is that it's classically in medicine brushed off because physicians don't know how to deal with it. They don't ask the questions because they don't really want to know, because they don't really have good therapies for it. And so people start to believe, they're told it's all in your head. Well, it is all in their head, but it's actually abnormal, right? So I'm just going to talk about some of the other stuff you can take out of these MRI pictures. This is more structure, so that was function. So the structure is abnormal too. So there was someone mentioning earlier about how they feel their bodies. I can't remember who asked that question. There's something that's called interoception, which is a fancy medical term, which just means how we kind of feel our bodies. So if you feel your heart racing, that's interoception, right? If you get nervous and you start to feel, I'm starting to sweat, and there's sweat dripping down your back, that's interoception. So it's how we feel and perceive our bodies. It's also how we feel and perceive our environment around us, but it's extremely important on how we interact with others and how we appreciate ourselves. And the two areas of the brain that are very important for interoception, one's called the insula, which is called the little human in many ways, or the emotional center, and the thalamus. So these are kind of like relay centers that are very important in the brain for helping us to understand our own bodies. Okay, that's what interoception is. So I'm going to just talk about some stuff here, and if I'm getting too, I'm going to try not to get too technical. If I do, just put up your hand, say you're getting too technical, and I'll rephrase it. So essentially, there's something called ALF, and it's not like a little furry creature that used to have a TV show. And essentially, what that does, it's actually these waves that reflect how the neurons in that part of the brain are working. Okay, so if you get a bigger wave, that means that's higher neural activity. If you get a smaller wave, so a narrower one, that's less. All right, that's really what ALF is. So you can extract this out of an MRI, and if you look in the insula and you look at ALF in the insula, people with PBC that have so this is a group of PBC patients versus a group of people without PBC who are healthy controlled. In their insula, they have lower neural activity. So I'm not sure what that means, but it's abnormal, right? It's not normal. And so there's something about, they've made a compensation within that part of their brain for dealing with how their body is communicating to their brain through the insula. Interestingly, in people that respond to URSO, I have no idea why this happens, their ALF normalizes. So there's something about responding and having that inflammation go down in your liver where that ALF then normalizes. Very good. Yeah? Yeah, all non-cirrhotic, because so we basically pick patients who are non-cirrhotic because when you start to deal with things like fatigue or any kind of brain changes, people always say, oh, that's just hepatic encephalopathy, which is the fancy term for people who have neural stuff with cirrhosis, right? And so to avoid that, we excluded people with cirrhosis on purpose. So these are all stage, like very early disease. You know, that's kind of cool. I have no idea why. I mean, what about other structures in the brain? So I mentioned thalamus is the other part of the brain. So the insula is one part of the brain that does this interoception, where the thalamus is probably the main relay station for all the signals that come from our body to our brain, not just interoceptive signals, but all the signals. So itch, pain, you name it, it goes through the thalamus. It's a major control center. And if you look here in the red, the PBC patients have their thalamus is smaller than the people who don't have PBC. So there's some change that's happening because of likely to regulate those signals somehow that are coming through the brain, coming from the body up to the brain, that's happening in all these PBC patients. So this is a consistent finding, right? The hippocampus is a major part of the brain that controls lots of different things, but it's really important for emotional control. And it's been known for a very long time that people who have major depressive disorder, so these are people that with diagnosed with major depression, their hippocampus shrinks. Okay. Yeah, so it's very important. Brain fog is very commonly experienced by people with liver disease and by people with chronic disease. It also, so I put brain fog here, it's really cognition, so it's how thinking clearly, I guess, and precisely, and so that it plays a major role in that. A lot of people with PBC, that's an issue, right? So when you look at patients with PBC, their hippocampus is smaller. And it's about 5% smaller. And so a lot of people say, oh, who cares about 5%? What does that mean? Well, actually 5% is what there's a huge literature published in major depressive disorder about a 5% change in the hippocampal size. So this is, and that's targeted with therapies in depression. So people use antidepressants and exercise therapy to increase the size of the hippocampus because it's the only place in our brain where we make new neurons. Okay, so I think there's also a potential to target that going forward. And that's what I think that maybe it was yesterday we were talking about exercise and things like that, but that's where exercise probably does play a role, because it increases a thing in our brain that's very important for generating new neurons. And so there's also, people talk about inflammation in the liver, but we also were very interested, is there inflammation in the brain? So when inflammation in the brain is called neuroinflammation, neuro is just essentially brain, and when we look at, when there's something called QSM, I'm not going to get into what that is, but it's essentially a way of looking at iron deposition in tissues, which is used in things like multiple sclerosis and Alzheimer's disease for defining inflammation in the brain. So it's becoming more and more used as a marker of inflammation in the brain. And what you can see here is that people with PBC have increased inflammation in their hippocampus. So these are all changes in the brain. So if people say it's all in your head, truly, as I mentioned, it's all in your head, and there's all kinds of stuff going on. I'm going to talk briefly about some other work that we've done in Calgary that I think is pretty neat too. So we were very interested to see if depression played a role in regulating PBC, because depression plays a big role in outcomes of many autoimmune diseases like IBD and things like that. So we actually went to this database in England. It's called the THIN database. It's about 12 million people that are in it, so it's a very robust database. And we searched, we pulled out, because you can take people that are diagnosed with PBC out of there, and we said, and you can also pull people with using specific coding, who's got depression. So we looked at depression, and we looked at so we looked at PBC patients who had a diagnosis of depression and who didn't. And we were fully expected that people with depression would do worse. This makes sense, right? I think most people would probably guess that. We found the opposite. So of course in science you can either ignore that and never publish, or you can dig deeper. So we dug deeper, because obviously it wasn't the depression. It had to be something it was something else. So we went through every medication, because all the medications everyone's on are listed. And a signal came out, and that was a drug called mirtazapine, which is an atypical antidepressant. Some of you may have heard of it. And essentially, all of the beneficial effect that we found was explained by mirtazapine. And people that were taking mirtazapine that didn't even have depression still got the benefit. And whether they were on URSO or not, they still got the benefit. So it was really quite fascinating. We published it. And so this is kind of the bottom line here. So essentially a hazard ratio is a way of defining statistically how much, where your risk would be, right? So essentially this represents almost a 75% reduction in outcomes that are hard. Like this is decompensation, so that's your liver packing it in, liver transplant, or death. Those are very hard endpoints, okay? There are no wishy-washiness in those. And so if you look at those results and you try to explain them in a broader sense, that if patients were using mirtazapine, that their median transplant-free survival would improve from 89 months to 134 months. And their 10-year decompensated cirrhosis rates would decline from almost 10% to 2.9%. So that's pretty neat, eh? And as I mentioned, the protective effect was independent of depression. So if they were on it for some other reason, a prescription of other antidepressants. So the other antidepressants didn't seem to have this benefit, and the use of URSO. So we had a lot of interest about that. Yeah. Is there any, what's that? There's side effects for everything. Yeah, so mirtazapine is quite widely used because it has a broad spectrum of activity, but almost all antidepressants, weight gain is an issue, almost all. And so that it'll increase appetite. And it can cause drowsiness. So often, actually, people are having issues with sleep. They use it at nighttime. So it's quite good for sleep. Those are the two biggest ones, yeah? Yeah, no, it doesn't have a lot. It's not. Dryness comes from anticholinergic effects often, and it doesn't have that. No, it's not an anticholinergic. It has quite a complex thing, but it interacts with multiple different receptors in our brain that do different things. And like a lot of antidepressants affect how serotonin is taken back up, so it increases the amounts of serotonin. This actually blocks different serotonin receptors and blocks a histamine type receptor. So it's quite unique. And the reason why I think it came out as a signal is because its biological activity is so unique.
Does it cope with fatigue? I mean, you're saying it's
I think if you took it in the morning, it would increase fatigue, okay? Because it would be, I don't know, if you take Reactin or something like that, which acts on as a histamine receptor, like I mean, you can get drowsiness from that. It's the same kind of thing. So you would take it at nighttime typically, right? And so when you wake up in the morning, the main effect of that fatigue is usually lessened quite dramatically.
So I guess we're talking quality versus quantity of life. Because if you're tired all the time and your brain isn't working, I don't care how long.
Yeah, well, you want both, right? Quality and quantity, right? And so, yeah, sure, but it's nice to have them both, right? And so, I think that obviously, I have a vested interest in fatigue, and I've been studying it for a long time. And so I'm very aware of how fatigue impacts people's quality of life. I'm sure everyone here is as well, and I know all of you are. And so I think that doing this sort of stuff allows us to get insight into things that we can do. And would it be mirtazapine? It may or may not be. I've not started to use mirtazapine in my practice because this is epidemiological data. But what I can say is we went back to the mice, and that's this, and we said, well, does mirtazapine do anything in the liver? And it actually does. It actually has quite an amazing effect on the immune system in the liver. It affects how a mouse's liver, affects how it responds to immune stimuli that damage the liver. So I think we believe this partly explains why there might be a beneficial effect with regards to the liver outcomes, right? Now the symptom outcomes, it would take a study, right? You'd have to actually give people that and see how. The thing is, mirtazapine is widely available, so it's very hard to do a study with something that's been around forever. This drug's been on the market for 20 years, right?
But you're not using it with your patients?
I haven't started, but I have a lot of patients that are asking me. I would never say, if they're on an antidepressant already and it's working, I tell them not to switch. Yeah, because, once you switch, sometimes if you go back, it doesn't work so well. So, but if it's not working well, then I certainly think it's a total option.